AOPs or quantitative AOPs, and advancing human relevant toxicology through data-driven models. So these webinars are about the collaboration between VHP4Safety and ONTOX and are organized in collaboration with Space Academy. I will briefly introduce each of the organisms responsible for this. So first, VHP4Safety, it's a Dutch research project that is funded by the NVA, the Dutch Research Agenda, and it's a program of the NVO. And it's coordinated by Utrecht University of Applied Sciences, and the RIVM, the Dutch Institute for Public Health, and running from June 2021 till May 2026. And ONTOX is a European research project, and it received funding under Horizon 2020 Research and Innovation Program, coordinated by the Vrije Universiteit Brussels, and running a bit in the same timelines between May 2021 and April 2026. And these projects both have a common goal of advancing human risk assessment without the use of animals or reducing the use of animals. So it made it a bit of a logical step to collaborate within projects. So there was a first meeting back in October 2022, and 6 collaboration groups were formed in 2023. So the QAOP group is one of these 6. And the Memorandum of Understanding was signed in February 2024, so last year. And this year we have decided to give the floor to each collaboration group. So this is the 3rd webinar. We already had the research output management back in February and stakeholder involvement in April. The recordings are available to watch on the YouTube channel of the SPS Academy. And for this one, we'll be talking about quantitative AOPs. So as I mentioned, each webinar focused on one collaboration group, and the goal is really to showcase both the work of these 2 projects and potential collaboration within the projects, but also to give the spotlight to early stage researchers to present their work. And we try to do them about every 2 months. The next one will most likely be at the end of September because summer break. And briefly about Space Academy, we're a network of early stage researchers developing new approaches, methodologies in toxicology, and we're trying to boost the careers of early stage researchers, giving specific training, either technical or general competencies. For instance, we have the summer school back in April, which was all about data and managing your data, visualizing it and communicating it. And yeah, we're trying to just give a space for young scientists to connect. So that I already mentioned, so various types of meetings, webinars, workshops. We're also trying to connect young or early career researcher to more established researcher or professionals. So if you're interested in joining us or in our events, we have a LinkedIn group. You can email us. Here are all the people that are involved in organizing these activities in the core group, and then we also have a board of members from each project of the ASPES consortium, which are the 3 projects, Risk Hunter, Precision Tox, and ONTOX. Now, today I realize I didn't change the title on this, but this is not Bridging Science and Society. This is actually quantitative AOPs. And we have 4 very nice early career researcher presenting with us today. We have Nathalie Dirks, who's a PhD candidate at Erasmus MC and the RIVM. We have David Paul, who's a postdoc researcher at Vrije Universiteit. Marvin Martens, who's a postdoc at Maastricht University, and Annika Drees, who's a PhD candidate at Vrije Universiteit Brussels. And for the first part, I will just give the word briefly to Marvin to introduce the collaboration group and what they're all about, and then we'll move on to Nathalie.
Thank you, Marie. So indeed, the collaboration group was established a couple of years ago and has had some collaboration meetings during official events, but also ad hoc, as mentioned in the slide. The last one being a little bit earlier this year. It has about 40 members in the email list, but indirectly more people or less people join those meetings. Depends how it gets forwarded. But it's a fairly large chunk of VHP that is covered within this within a collaboration group. The leaders is not just me, it's also Eliska. She could unfortunately not make it for today's webinar. So she would be from ONTOX side, the collaboration group lead, and myself from VHP4Safety. We have a couple of topics that we seek collaboration with or discuss about, which is the both qualitative and quantitative AOPs, because both projects are working on both. Also quite a lot on molecular aspects, so the molecular pathways, molecular AOPs, SVGN models, and the overlapping case study specifically out of the multiple in both projects is the kidney case study with also people that are part of both projects. So that's the one where we see most directly a connection between the two projects. And then I give the word back to Marie.
Thank you. So Natalie, you can now get started with your presentation. For people watching us, you can just ask questions in the Q&A if you don't want to raise your hand or unmute yourself or whatever, but we will address the questions after each presentations all together. But feel free to just send questions in Q&A during the presentations.
Okay. Okay, so thank you for the introduction. Today I will tell you a little bit about what I'm doing within the VHP4Safety platform consortium, which is the development of a testing batteries to assess the developmental neurotoxic effects of chemicals via disrupted thyroid hormone homeostasis, which is a lot to take in at once. I'm going to get you through it one by one. So first VHP4Safety. In short, we're building a virtual platform together, and in the end, the goal is to have safety assessment of chemicals based on human data. And today I will focus a little bit on clinical data, physiological data, and in vitro models. And this is our project approach. We have different research line building and implementing and feeding our platform. And my focus is work package 2.4, which is the Asian gender case study focusing on thyroid hormones. So why is this important? Well, we know that if a mother is hypothyroid, so if she does not produce enough thyroid hormones by herself, later on in the child, we can see different adverse outcomes. For example, this can be a lower IQ, decreased motor function, and also, for example, growth retardation or hearing loss in very severe cases. And the condition of hypothyroidism can happen spontaneously, but also, of course, different compounds can have an impact on the thyroid hormone balance. And in the news, we see endocrine disrupting chemicals everywhere. We hear a lot about PFAS now, for example, in the Netherlands with the chickens, which you have at home, that their eggs have more PFAS than we buy them in the store. And there are more examples in a lot of different newspapers on EDCs, so endocrine disrupting chemicals. And with the current testing guidelines, we're working with sort of a black box model. So we expose an animal, something happens within this black box, and in the end, you see the adverse outcomes. This can be anything, for example, liver cirrhosis, tumor formation, anything can be scored. However, our goal is, of course, to use animal-free experiments. And the goal is to predict the adverse outcome that would happen by defining the mechanism of action of a chemical. And this is actually where AOPs come into play. So an AOP, or an adverse outcome pathway, is a structured representation of biological events leading to an adverse outcome. You can see it here in the bottom. An AOP is initiated by a stressor, in this case a compound, which starts at the molecular initiating event. This is the start of the whole cascade towards the adverse outcome. And between where the compound targets and our adverse outcome in the end, we have key events, so key Es, which are measurable essential steps along the whole pathway towards the adverse outcome. And then to build our AOP, we need some background information. So especially for us, of course, the thyroid hormone homeostasis is very important. And in short, we know that in the hypothalamus, thyrotrophin releasing hormone, or TRH, is being produced. This stimulates the pituitary to produce thyroid stimulating hormone, which in turn stimulates the thyroid to produce either T3 or T4. T3 is the active hormone, T4 is the inactive hormone. And when distributed through the body, they can be taken up by the thyroid hormone transporters, which can be MCTs, so monocarboxylate transporters, OATPs, or for example, also lactic acid transporters. Within the cells, T3 and T4 can be metabolized by the deioneases. These are the enzymes that convert the active hormone back into an inactive form, or the inactive one into the active form. So it's really important. And you have different types of deiodenases, but deiodenase type 2 and type 3 are the most important in the brain, so D2 and D3. And for the transporters, a very important transporter for thyroid hormone in the brain is MCT8. It can transport both T3 and T4, and it's mainly present in brain cells and especially at the blood-brain barrier. And we know from a clinical case that if there's MCT8 deficiency, so a disease where a person does not have enough MCT8, so not enough of the transporter, during development, not enough T3 will enter the brain, and we can see some, very bad adverse outcomes in the children. So here you see a clinical case of MCT8 deficiency, and in this case, you see the myelination of axons, an axon. Over here, you see the dark purple line, which is the myelin surrounding the axon of a neuron. And then in an MCT8 deficient fetus, you can see that there's no myelination at all. So similar part of the brain, no myelination visible compared to a normal child where there is myelination. Same for example for synaptophysin. Here you can see all the dark spots surrounding a Purkinje cell, which are the synaptic boutons. And here you can see the vague tracing of the Purkinje cell, but nowhere near as much synaptic boutons as in a normal situation. And now our goal was to incorporate all of this into an AOP. So we're thinking, okay, we know thyroid hormone, brain development, what can go wrong? Then we come to MCT8 deficiency, which actually shows the causal relationship between thyroid hormone deficiency and the neurological outcomes. We needed to choose our adverse outcomes, which were in this case cognition and motor function, also both seen in the MCT8 deficient patients to be either decreased or almost completely lacking. And then our goal was also to incorporate human physiology, as we not only wanted to show what happens in the brain, but also which kind of cells. So this is the AOP as has been published this year. It's a lot of information at once, but in the gray area, you can see the fetal brain, and we have 3 different cell types. So we have astrocytes, neurons, and oligodendrocytes, all having their own function within our brain. In green, you can see all our MIEs, so molecular initiating events. We have our transporters, the MCT8, the OTPs, and also others. We have our deiodenases over here, D2, D3 over here. And then also the thyroid hormone receptor, which when T3 binds to the receptor, it gives, promotes the whole downwards stream effect of the thyroid hormone. And we can see here, for example, that when there's less T3 in the neurons, you can see that there's decreased synaptogenesis, decreased network formation, and neural network communication, which is also what we saw in the brain pictures a few slides back, where there was almost no synaptic bouton, so a lot less communication. Also in the oligodendrocytes, which are responsible for the myelin surrounding the axons of the neurons, we know that if there is less myelin, the insulation goes down, and then also there is less neural network communication possible. And in the end, all this can lead to a decreased cognition and decreased motor function in the child. And since we want to use AOPs to build an animal-free or human-based testing battery, we decided to take different parts of the AOP and try to build assays in the lab to mimic these parts. So we have our first tier, which are the molecular initiating events, and this one focuses on our transporters, deiodenases, and the thyroid hormone receptor. The second part focuses on neural differentiation, so it really takes into account a later part of the AOP and also a later stage of development. So for our 2 tiers, we have for the first tier different cell lines which represent the different cell types of the brain, in which we look at the thyroid hormone transport, deadenation, and the receptor. And in the second tier, we look at the model for neuronal differentiation, where at this point we look at gene expression related to neuronal differentiation, and we're working to set up microelectrode arrays as a functional readout for the neural network activity, so actually measure the communication between the cells. So for the first tier, this is a very schematic overview of our cell. So we culture 3 types of cells, the astrocytes, the neurons, and the oligodendrocytes. And for all these cell types, we wanted to see if they could either take up thyroid hormone, because if they cannot and they don't have any transporters, it's not a good model to start with. Then do they have the deodenases that they should have according to literature? And in the end, do they have the thyroid hormone receptor? And also, can we activate it and measure that? So for the first part, the thyroid hormone transport, you can see here the uptake of thyroid hormone in our 3 different cell types. You can see over time from 0 to 60 minutes in all 3 cell types, our thyroid hormone uptake increases. So actually the accumulation is building up. And we can see that for both T3 and T4. So we know all our cells have transporters for both T3 and T4. So then the question was, can we also inhibit this uptake? And here you see for the same cell types, the percentage of uptake compared to a control if we would either add silicristin, which is an MCT8 inhibitor, or lesinurad, a more general OATP inhibitor. And especially for the astrocytes and the oligodendrocytes, you can see here that both T3 and T4 are significantly inhibited compared to our controls. So we know they have MCT8. Also for the neurons, probably the ratio of MCT8 compared to other transporters is less, but we can still inhibit it. And also OTP is in all 3 cell types significantly inhibited. So now we know our cells have transporters, and we can actually measure if a compound inhibits the thyroid hormone transport. Then the deodenation. We know from literature that astrocytes are supposed to have D2 because they use the inactive form T4 and metabolize it into T3, which is the active form. And one of the goals, or the astrocytes want to present the T3 to the neurons, which they can take up again. And the neurons and oligodendrocytes normally have D3 because they need to strictly regulate how much T3 is actually being in the cell and ending up at the receptor. And these graphs are from our own lab, where we checked whether or not our cells had the corresponding deodenases. And you can see here that the astrocytes actually have D2, and the oligodendrocytes and neurons have D3. The other deodenases were not found in the other cell types. So this corresponds with what we hoped to find. And then we checked with a reference compound, can we actually measure the inhibition of our deodenases? And you can see for both the oligodendrocytes and the neurons, both ipanoic acid, AOP, and xanthomol, which are both reference compounds for D2 and D3, can actively inhibit the deodenation. But this is a cell lysate, and our goal is to keep the cells intact, to have a little bit more of the sort of endogenous surroundings, because then you also take into account the uptake. So we also tried the whole cell metabolism assay, where we kept our cells intact. In this case, I'm going to show the oligodendrocyte example. So we can see here, this is the picture of the cell lysate. Ipanoic acid inhibits clearly. Here you see the ratio of the different metabolites. So we have a lot of T3 in our control, a little bit of T2, and a lot of T1, which means that a lot of T3 has been converted by D3 into a inactive metabolite. And then we have 4 concentrations of ipanoic acid. The highest concentration, there's almost directly comparable to our tracer. So there's almost no T3 being converted, meaning D3, the diodine, is almost completely blocked. And the lower the concentration, the more green and purple we see, meaning that the inhibition is less, and you see a nice dose response from ipanoic acid. However, when we look at xanthomol, on the left again, very clear response when you have the cell lysate. However, when we keep the cells intact, you can see that all the different concentrations do not really differ from our control. So this was very interesting because we assumed that when it would inhibit the deodenase, you would see it in whole cell metabolism. However, this shows that it's a little bit more complex than that. So probably xanthomol does not even enter the cell, so it will never reach the deodenases. So it can also not inhibit the deodenases. So it's very important to keep in mind when we're developing the assays to compare and see what's really necessary to measure and do we see the whole picture. Then for the receptor activation, we tested a dose response for T3 to see if we can actually measure thyroid hormone receptor activation. On the right, you can see our dose response from 0 to 1,000 picomolar, and you can see a nice increase of our receptor activation, which is KLF9 gene expression. And the goal was to find the optimal concentration to use together with our endocrine disruptors to see whether if we add a compound, you actually see more activation of the receptor, so you have an agonist, or maybe antagonistic properties when you see a decrease in activity. Well, we're still working on that. And that was actually the first tier, so the 3 cell models with the 3 different assays. And then for tier 2, we're working on the neural differentiation, where we're working with human stem cells. This is based on the protocol of Victoria de Leeuw, also working at RVM, who set up stem cell differentiation into, in the end, a neuroglia co-culture. So the H9 stem cell line is being cultured for 19 to 26 days into NPCs, neuroprogenitor cells. And that is the stage where we're actually going to expose these cells to our compounds. So we keep them intact and normal for the first part of the differentiation. And when they're neuroprogenitor cells and actually need to start differentiating into neurons and astrocytes, that's where we add our T3 and our compounds. So our goal in our plans actually are to add the compounds during the MPC to differentiation stage with and without T3 to see the different effects. And then also add different kind of compounds to see what the effect would be on the neural differentiation. So we're going to add silicristin, which is the MC8 inhibitor, xanthomol, which was the D3 inhibitor, but we did not see that in the metabolism, but maybe we can see something here. TBPA, which is a brominated flame retardant, chlorpyrifos, pesticide, NH3, which is our control for the thyroid hormone receptor antagonist, and MBP. And the idea is to use 2 concentrations to see if that also would have a different effect and measure different genes also related to neuronal differentiation. But we're still working on that, so I'm not going to show any data on that yet. So to conclude, for tier 1, our chosen cell lines are suitable to measure thyroid hormone transport inhibition. We have the deodenases present in the corresponding cell types, and we can also measure the inhibition when we keep the cells intact. And we can measure the activation of the thyroid hormone receptor and also the inhibition if needed. For tier 2, as I said, we're still working on that. We will measure the effect of endocrine disrupting chemicals on the differentiation. And we want to expand our readout with a microelectrode array to also hopefully measure the effect on the neural network activity. And with that, I would thank you all for listening, and also want to thank my supervisors for guiding me through this whole process. And of course, VHP4Safety for making this all possible. Thank you.
Thanks, Natalie. Very nice presentation. Do we have any questions of the audience? I haven't seen anything in the Q&A or in the chat yet.
Just very clear.
Oh, there is a question. In the AOP, increased activity overexpression of D3 is regarded as an MIE, but not affected by T3, T4. How is this process undergone?
The AOP. So D3 is... Sorry, I need to... Can I read the question?
I don't know.
At the bottom of your screen, there should be a Q&A
button. Yeah, sorry. Okay, if I send it correctly, we do consider the effect of a compound on the deodenases as an MIE because that is the first place where a compound can strike, so to say. But it's not directly affected by T3 or T4 in this case, because we assume that within the cell, if the compound affects a deodenase, that then the level of T3 or T4 is affected. So we take, in that order, we take deodenases first, and then T3 and T4 as a secondary event, instead of T3 and T4 influencing the deodenase beforehand, which is also happening in real life, but we didn't include that part in our AOP.
Right, thanks.
That answers it.
I hope this answers the question for whoever asked it. If not, just raise your hand or give us a signal. Okay, yes, that answers the question. Is there any other pressing question? Juliet, thanks you for the great presentation, and asks how you couple this with human data. And also to the organizers, is it possible to see who is in the audience? So we, as organizers, can see it. I don't think the attendees can see it. There's 40 participants right now. Hope that helps. And now, Natalie, can you couple this with human data?
Yeah. Well, we're currently trying to figure that out together with Filippo and Joost, who are also working in VHP4Safety, because there is a paper on MCT8 deficient patients and their phenotype depending on the MCT8 mutant genotype. So we can see that in that paper, they show that the severity of the mutation does matter for, in the end, the outcome on motor function, for example. So now we're working together to see if it's possible to connect those data, for example, to our the QAOP in development. But we have to be really careful because you cannot just say, oh, my compound inhibits 20%, so that means in the end 20% decreased motor function. So now we're trying to figure out, also together with the collaborators at Rotterdam, at the EMC, to see how can we safely incorporate all of this into our QAOP.
Right. Hope this answers your question, Juliet. And if not, again, raise your hand or give us a sign. We have another question / comment from Chris. Strictly technically, that makes it another AOP. So what you have is technically is really an AOP network?
Yes. Yeah, correct. Thank you, Chris.
Okay. All right. Are there any other questions? Pressing. I don't see anything right now. Then we can move on to David.
Hello. Okay, let's get this going.
Thank you, Natalie, again.
And then 1 second, I am sorry about this. There we go. All right. So, hello everybody. I'm here to talk about using in silico docking and quantitative AOPs. In this case, we're going to be talking mostly about the AOP that Natalie just presented on. So there will be a little bit of redundancy on the biology as well as the overall mechanism of the action. So, as a small introduction, I work at the Vrije Universiteit Amsterdam in the Molecular and Computational Toxicology group. And specifically, we are a small group of biomolecular simulation, a subgroup even, involving Dan Greke, this tall guy in the back, as well as myself. And we do biomolecular simulations on toxicology relevant protein-ligand interactions. In the case of an AOP, which now I cannot advance slides, there we go. In the case of an AOP, as Natalie explained, we have a molecule that interacts as a molecular initiating event with a protein to form a complex. This leads to a series of key events, which generally leads to medical outcomes. As a chemist, as a computational chemist, the part that I can really approach and I try to deal with more is the molecular initiating event. And this involves looking at how a molecule interacts to form a protein complex, which then would subsequently initiate the events. To do this, we use a tool called molecular docking, in which it takes an arbitrary molecule and an arbitrary protein, and these are just figures for a reason, undergoes a process of pose searching and pose optimization to form and rate and look at protein complexes. This is done by the help of a scoring function, and this scoring function is an arbitrary bit of math to tell and rate how the poses are using physical forces or empirical measurements based on crystal structures and known data. From this, we get a series of binding energies, and that I measure in kcals per mole because I'm still an American. And we have strong binders, which can present an increasing risk in your AOP, as it will bind stronger and thus inhibit more, to less inhibiting molecules, which are no risk at all. And this allows us to classify and sort of coordinate what molecules may be a risk for particular MIE and a particular AOP of interest. In the case of this, we're going to look at MCT8. The one requirement of this is that the molecules and the proteins themselves must be well defined. We cannot take keratin or mixtures of molecules, and the protein has to have a known structure. Also, it has to be a single molecule. It has to be we have to know some ligands to be able to test things. And lastly, it has to interact non-covalently, which to some importance, it negates the idea of metal interactions. So we cannot look at particular metal interactions like cisplatin, but we can do things that interact primarily in a drug-like manner through hydrogen bonds, van der Waal, or ion pairing interactions. And this is the part that we really want to look at. For this, we really want to study out into the thyroid toxicity, as Nathalie presented, and we're looking in particular at MCT8 because this is the more difficult, less known molecule. As Nathalie already introduced, MCT8 is a membrane-bound thyroid hormone transporter. It transports a variety of monocarboxylate ions through its transmembrane domain, and this plays a role in the fetal development in terms of receiving thyroid hormones from the mother into the baby during the first 10 weeks of pregnancy. It has been reported that silymarin extracts are very commonly used throughout Germany and Europe as a folk medicine to heal liver injury, which has been somewhat substantiated. And this is relevant because silymarin extracts contain 20% by mass reported MCT8 inhibitors. So if we look at the flavonoid content of this milk thistle oil, it contains a large dose, an incredibly large dose of MCT8 inhibitors in the form of silicristin. The other molecules themselves are very likely MCT8 inhibitors because of their chemical similarity as well. So it's a very inhibiting mixture. It's very toxic in this case. And so to looking at the system we're going to look at molecules, how they interact with proteins, and this infers the hazard through the AOP that Nathalie has generated in terms of what molecules may evoke the same MIE and thus the same key events which lead to adverse outcomes. The real challenge of this, of course, is going to be that MCT8, because it's only of toxicological interest so far, does not have the same sort of supporting data in terms of crystal structures, and that we'll have to deal with as a challenge. So what we're going to do is we're going to predict the structure of MCT8. We're going to take a larger library of inhibitors or potential inhibitors, screen them with molecular docking, and identify natural products that could be inhibitors for consumer goods. For known inhibitors, we actually have screened the literature, and we have different strengths of known inhibitors. This is a very ideal case for us where we have strong inhibitors, which are sub-micromolar, medium, which are sub-tens of micromolar, and weak inhibitors, which are sub-millimolar inhibitors that we can use as control samples to sort of give a scale of danger between yellow and red of how a compound may inhibit MCT8. In addition, we also have some non-inhibitors. In the case, we have substrates like the thyroid hormones themselves and other medicines that have been well studied, as well as potential inhibitors that are studied, which were found to be non-inhibitory, such as tryptophan, caffeine, and various other compounds. Triac appears twice by mistake. So together we have a very nice library of known substances, which we're able to then classify our MCT8 binding affinity for unknown ligands. As for the structure, if you just try to alpha-fold MCT8 itself, you'll get something that resembles MCT1, which is the only crystal structure for a monocarboxylate transporter. And the problem with this is that the binding site, which is located up here on the top view of the best model, is always closed. And we can see that doesn't really work out for binding. If you try docking these models, you'll get erroneous docking into this open site on the bottom, which is not where actual binding occurs. That's the transportation exit into the cell. So this is, if you just do a naive look at AlphaFold3, you do not actually get a useful binding site for docking, and thus you'll come up with completely erroneous predictions for MCT8. What we found, though, is we can trick AlphaFold3 if we give it instead a AMP ligand, which is one of the few ligands that's by default built into the system. It will place the anionic ligand with the phosphates in the same site that you'd expect a carboxylate ion to be. And there, you can also nicely do these phosphorylations, which forms the loop structures of the external loops correctly. So we actually get a much better model of what we would expect MCT8 to look like. Taking this, we took this a step further. This model, we generated 5 models, and we want to then validate them to use in our approach for identifying MCT8 inhibitors. So we took our nice our library of inhibitors, we screened them with docking, and we got a series of scores where the lower the score, the better the binding, and thus the more toxic it is. And we beautifully see that the substrates and negative inhibitors score higher and thus are less dangerous. Whereas with increasing danger or increasingly known inhibition, we get a lower docking score indicating stronger binding. So this was a very good sign that we have a method of going between strong and weak inhibitors. Considering that AlphaFold generates more than one model, we also looked at the other conformations generated, and we found that inward open conformers, like the one that was generated in the first slide, actually do not portray any sort of useful predictions. In fact, they predict that the substrates and the known negatives would bind stronger than known positives inhibitors, and that these guys would not bind at all. So we have a really good method of validating the protein structures we get to be an accurate or useful model, although not so much accurate, but more useful means of determining or distinguishing inhibitors from a general pool of ligands. And that's exactly what we did next. We started expanding the search. Instead of looking at just the known molecules, we started expanding out, looking at similar molecules, i.e. flavonoligands. And for this, we just did a simple Wikipedia search of all molecules labeled flavonoligands. And there's 23 of them, which were then labeled and found to be relatively strong inhibitors compared to what we know from silicristin and silymarin extracts. And for this, we're just comparing positive to negative as a relative way of triaging danger. Other sources of flavonoligands perform similarly, showing that generally, as a class of molecule, flavonoligands are dangerous. And then going on beyond that, we screened flavins with increasing numbers of molecules, finding that these are good medium inhibitors, and then also a diverse library of similar ligands generated by structural similarity searches. So as a sort of model, we can see that there is a strong bias to finding molecules that are good inhibitors based on structural similarity using molecular docking, which is more or less exactly what we expect. You can also see here that we're screening increasingly large libraries up to 970. So this only took about 10 minutes to time to actually do the docking. And so with this sort of motivation, finding that similar molecules behave as we expect, we started screening industrial relatively large libraries of natural products from herbal remedies, including licorice or drop, red clover, fenugreek, maca, and these sorts of things that were up and coming, more sought after, more widely used, or more widely available products in terms of the herbal product market. From these, we find that actually a lot of herbal products contain what could be strong MCT8 inhibitors, raising a lot of red alarms for bells, although a lot of these compounds themselves would be very low abundance inside these herbal medicines, in that silymarin is a bit unusual in the fact that it contains mostly MCT8 inhibitors and that they are, for the better part, 80% of its mass. Only exceptions are bacopa and drop, both of which are already display other signs of fetal toxicity as aborphicants. So not the most useful findings, but it's a demonstration that we can show that through docking, we could look at large libraries of natural products. So in review, molecular docking is a really nice tool for at least screening the MIEs in terms of looking at AOPs to identify new molecules of interest that might be useful for toxicological evaluation. The predicted protein structures can, in fact, be used in molecular docking, even from AlphaFold. but they must be rigorously validated using libraries of known molecules, in which requires a certain minimum amount of data to build off of. And lastly, that docking can screen thousands of molecules with automation. So we can really expand this sort of approach to develop large-scale screenings without needs of animal testing or other high-throughput screening approaches. And lastly, that this can be used as a method to triage molecules to at least sort out what products and or what molecules themselves might be needed to screen. And with that, I thank you all for your attention, and I'd appreciate any questions.
All right. Thanks, David. Any question from the audience? I see there are some. Okay. Susanna is asking, well, thanking you for your presentation. I'm not sure that's for David, I think. I can understand that you can predict the potential affinity to MCT8, but in reality, this will depend how much the chemical reaches the target site. Do you plan to incorporate the toxicokinetics and QAOP? Even within the in vitro system, it can be that the chemicals have different fractions available, which will result in effective different potency. For example, the same characteristic that lead to good affinity to MCT8 can also lead to higher unspecific binding to cell lipids and proteins.
Yes, that is a good question. Thank you very much, Susanna, for that. It would be nice to integrate this to toxicokinetics to the QAOPs. Not exactly sure the method of doing that in terms of an automated sort of toxicokinetic model. If we could get accurate concentrations for exposure this way, that would be ideal. I have looked into available data in terms of Silomar and exposure through teas or oils, and therefore the levels that we'd expect for concentrations. And we could try to do that in the future. But that would require a lot of establishing work, and I think that would be a nice lofty goal for the VHP or the ONTOX to have. Non-specific binding with other proteins, however, would be impossible to screen, as we do not have a full breadth of all available proteins in the human body and all possible conjugates thereof that would create potential binding sites. So for that, we should really rely on the precautionary principle, wherein we look at molecules that could be dangerous and treat those as a potential source of hazard for further testing with cell culture methods or in vitro approaches, rather than in silico.
Right, thanks. You have another question from Yulen. Thanks for the presentation, very nice, naive questions. So 2 of them. Is AMP needed to activate MCT8 in the human? And is your model is constructed with 20 to 30 chemicals? Is this potent enough?
Oh, thanks, Julian, for that. These are both good questions. On the first one, AMP here is just being used to trick AlphaFold and to give me a model that has a ligand inside. AlphaFold itself will predict only a certain number of biomolecular ligands, and for I guess for drug research reasons, they have not made it available to include arbitrary ligands inside the model, or at least at the time I did this study. So this was a sort of trick to include an anionic ligand that would be placed in the correct position, which can be used to identify a docking site for molecular docking and give an overall ligand-bound conformation of the molecule. It's actually, biologically, AMP does not interact at all with MCT8, as far as I know. And this is just more of a trick to trick the computer to looking for carboxylate or phosphate to put in that site. As far as the 20 or so chemicals we use to validate this model, it's a bit of a question of, is it enough? Well, there's always that question, right, of what level is enough? And more is always better to a certain point. And I would say it would be nice to have more exemplars to use with known inhibition. But right now in my pockets, in the literature, there's a very small range of chemicals which have been shown for MCT8 inhibition. And this is sort of a limited sort of data set to work with. So for what we have right now, it seems to work fairly well. And we hope to use it to identify more examples of inhibitors to then expand out the model, which would be done in collaboration with Nathalie, Edward, and Marcel as well. Because that's the hope.
Right, thanks. Then we have actually some questions in the chat from Ivo. How can we be sure that the docking you predict is actually inhibition? Could the chemical bind to a hormone instead and be inhibited?
How can we know the chemical? Sorry, can you repeat that one more time?
How can we be sure that the docking you predict is actually inhibition? Could the chemical bind to a hormone instead and be inhibited? And then he said, I mean the chemical bind to the abstract of the receptor.
Okay, so in this case, the inhibitors have been screened through several different model approaches, including direct absorption-desorption studies, so we know that it's not binding to an internal pocket. So we have a lot of good idea that the molecules are screened as strong, medium, or weak inhibitors are, in fact, strong, medium, weak inhibitors, and not going through some other means, such as binding through D3 or D4, and not being lost inside the cell, essentially. So we're pretty sure that those are good starting points. And the second part of that question, sorry, since it's here. Sorry, I'm just pulling up the chat myself to read the question. The chemical could bind part of the hormone, and we're not screening that at all. No, if we have some sort of conjugation occurring, such as a protein or some sort of protein in the bloodstream, like albumin binding the hormone, then we couldn't possibly assess that. Again, that gets to the idea that we have to know every single constant of the human body, or the bodies in question, or all human bodies, and that's just unreasonable to try to expect to have any screening on. So we really have to narrow our question to one protein MIE, one protein molecule MIE, and that's it. So this is, yes, a limitation of docking in that sense.
Right. Okay, we have 2 more questions in the chat and 1 more question from Ivo, but I see we're already a bit running out of time. So if that's okay, I will collect the questions and send them to David.
I could also answer them directly through the Q&A here and just type an answer if that's okay. I think that's no problem. Or send it as an email is fine.
Okay, then maybe if you can answer them as Q&A directly, and then we move on to Marvin so that we can get to finish a bit on time.
I will be quick. Thank you.
It's not to rush you, Marvin.
No, but I also know that there's the last speaker after me still, so it does need a bit of rush. But yeah, so yeah, thank you for inviting us to present here. I'm the last one from VHP to present, and I'm very sorry for not using the shared common layout. I realized this too late. So I'm Marvin Martens from Maastricht University, Department of Translational Genomics. I'm happy here to present a bit of the other types of dynamic tools and analysis that we're doing in the project. We saw now the in vitro and the AOP development, really nice introduction on that, and I'm now following up on that and starting to integrate it with transcriptomics data as one of the case studies that we used. Scrolling a bit through it, we know that in toxicology we have a whole array of biomarkers to assess whether some effect is happening or whether some disturbance of processes is occurring or on different levels of biology. But the ones that we're also very interested in this project and also in our group is the omics type of data sets. So think of the metabolomics, proteomics, but specifically here I'm talking about transcriptomics. And we want to assess what we can do with it. We have known from literature, from many applications before, that omics data can be used to identify many different things and assess many different types of effects. Overview here, not going through it. But the question then arises, can we use transcriptional activity as a biomarker for toxicity? And then aiming towards key events, for example. In the risk assessment, so thinking about the application of transcriptomics in risk assessment, we see kind of a dilemma occurring that in the scientific field, they are used for many different types of applications to understand modes of action, to assess whether certain processes can be inhibited or activated by small interactions, understanding the biology. But on the regulatory side, there's a lot of holdback. There's a lot of issues that are involved in the consistency, the general acceptance of certain protocols, agreements on how to process and analyze and handle the data listed here and also reviewed in a couple of papers that make the application really complicated. So complicated that there's papers out there, even in sets of 3, that describe all the current status of of how data, transcriptomic data, in the field of toxicogenomics is being well designed, generated, used, and analyzed and interpreted. In this presentation, I'm mostly focusing on this last section, the interpretation, especially in regards of the adverse outcome pathway, highlighted there in the right side. From the AOP wiki, the main source of AOPs at this moment, we see that over 90% of all the key events fall under the molecular, cellular to organ level events, and have also a high likelihood that we can support them with molecular process or describe them based on the mechanism of action that is underlying it, the active pathways or response pathways. So what do we actually want to do with this? So the idea to link key events to gene sets would allow us to perform analysis with these type of transcriptomic data. Especially here, the middle question is then relevant for us. So which set of biomarkers indicates a poor prognosis or good therapeutic response? This is one of the applications of transcriptomics in the field already. But then the question arises, how do we define gene sets per key event? Key event are textual descriptions, often supported by a lot of literature, as we saw with Natalie's presentation. Also on the other side, for gene sets, we already have so many data sets that define gene sets, either by association, by certain types of different evidences, but also based on molecular processes that we do understand, such as pathways or other fairly well-known resources such as gene ontology or other ontologies. Also in the project, as a side note, we have also TXG Mapper, which is also mapping key events, gene sets to key events using maybe a gCNA and then enrichment. We have a lot of options to analyze and a lot of possibilities to analyze and set parameters to perform our data interpretation. In our project, we are aiming for wiki pathways. This is a pathway that we maintain in our department, and is a molecular pathway database that is mostly community driven and then also really flexible to work together with tools. That's also what I take advantage of here. So generally, the structure looks like this. This is an illustration. On the top, we see the AOP wiki with its textual descriptions, textual annotation, and some minor ontology tags. I will not go into that. And in the middle, we see kind of the connective model, how the key events in the AOP wiki would be connected to molecular pathways and wiki pathways. And this model becomes an interactive tool. This is a software that you can load into Cytoscape or into PathVisio, our molecular pathway drawing and visualization tool. And we can also then, of course, go further and see what are all the genes and processes and how genes in these processes interact with each other to result into some downstream effect. And for that we can use the transcriptomic data. Again, here summarized, and on the right side we see one of the examples of the project. This is the Parkinsonian motor deficit AOP, not the case study that Natalie is doing, but the one of Julia. And this one is on neurological adversities with retrotinone as one of the stresses. It has been a fairly well-known AOP in the field. That's why this illustration. And this connection allows us to, perform some enrichment analysis, overrepresentation analysis, using transcriptomic data of all origins. To illustrate a little bit the case study, this is going to continue from where Natalie ended with the AOP. I wanted to use transcriptomic data to assess whether key events that are part of that AOP would be activated or not. We were given a data set from the RFVM. This data set is an RNA sequencing data set. It's also pre-processed and quality control by colleagues at RFVM from the project. It's that model that Natalie already introduced, where there's levels of T3 exposure at 3 different conditions and 8 replicates, and differentiation process ongoing. In general, there's two comparisons being made between physiological level thyroid hormone 4.1 nanomolar versus a lack of T3 or a high level of T3 being 100 times or really zero, well, not detectable, so it's a low T3 still. Of course, with multiple testing correction. On the right side, we see the amount of genes that become significantly altered when you have these two different situations. And you can clearly see that with a high T3, there's a very low number of genes being significantly affected. Or with a lack of thyroid or with a low thyroid, there's almost 3,000 genes being affected. The table shows the top regulated genes, not going into detail with that, but I want to indicate this KLF9. This was one of the genes that Natalie mentioned, is a marker of the high level T3. So with increased T3, you assume also this one to increase, and that's also the one that seems to be the strongest upregulated in high T3, but downregulated in low T3. So that confirms from the RNA sequencing data what's happening on that level. I need to check what's the time actually. Okay, then I think I'm okay. So then we started mapping together with Natalie and with Ellen the molecular pathways onto the AOP that was developed by Natalie. I tried to visually make it similar so it's easy to interpret. And as you see for most of the key events, or some of the key events, you see the green boxes. These are then the molecular pathways that are in wiki pathways that we then associate with the key events that we see in the AOP. There are some challenges with this, that the processes and the dynamics in the biology are very complicated. It's of course differentiating cells in very early stages, so there's a lot of things happening in different times. And also the pathways that we see here are not per se the pathways under the situation in the context of neurodevelopment. They are more generic and general processes that we know from many different sources of origin, information sources. So there's a bit of translation to be made. Also, as you saw, this is not directly showing the effect of silacristin, this mixture that is being studied, but it's mimicking the effect of a low thyroid situation. So I'm not looking here at the MIE activation specifically, looking more at the available T3, T4 before they transport within the brain and within the cells. When visualizing this and taking some of the statistics out of this from via cytoscape, we can see on the top right side with the high T3, we only see one single gene related to this process being highly upregulated. I think this is actually I cannot read it. One of the genes that you saw strongly upregulated in both data sets. And on the bottom left side, we see the low T3 affecting very strongly the cell cycle, seeing mostly a downregulated pattern, and also the BDNF pathway. The one that we saw is the first key event after low T3 activation. And if we calculate the odds ratio, so the overrepresentation analysis, we can see that these three processes connected to the low T3 have a statistical support. They are statistically different when compared to the physiological situation, which would be interesting to explore further. So from here, we can actually go into the pathways that are kind of behind this model, and we can see how the pattern of gene expression, displays itself over the molecular process. We see some regulations on the top at the receptors of BDNF, but also we see many of the important key kind of junctions within the pathway that are involved in the downstream signaling. We see patterns of upregulation and patterns of downregulation, leading to different effects downstream. Of course, there's a lot of things to interpret from here, so I will not go into it. But it gives us a fairly good idea what kind of processes or what kind of downstream effects are being more active than with a normal physiological level of thyroid hormone and which process would be inhibited. If going into gene ontology, this is also inside the script with Klugo, we can check if we actually found with the molecular AOP many of the involved processes to be confirmed. And we did see for the majority neurological neurogenesis related processes, especially in the upregulated genes. However, sorry, I actually mixed them up. This is downregulated, this is upregulated. Sorry for that, I should fix that. But we saw that with the downregulated, that there's also many different processes being missed or being dysregulated, such as in metabolic reprogramming. There's a reliance on a different fuel source according to the gene expression of enzymes that we see involved in those. So it also shows very clearly some side processes that might not be causally linked to the outcome, but are still essential to understand the biology and what's really happening. Currently, we're still trying to understand most of those changes and support them with the literature that is already available on neurodevelopment, but we are not completely finished with it yet. So a short recap of linking the omics to AOP via pathways. It allows us to explore the biology in a much more detailed way. Just in general, having the pathway model is intuitive to read, besides having a full text describing the processes. And for us in bioinformatics, we like to know the details and the small interconnection between genes and processes and proteins, which we can then also have. We can use transcriptomic data to assess activity or assess dysregulation of early key events. However, there are some limitations if we can see if things are being activated or not activated. That requires still a human interpretation of the pathways processes. There's also a challenge regarding subtle or non-specific processes, and for example, the interplay of causative and responsive pathways that are maybe not directly connected to a key event, but might be slightly upstream or downstream, which would be kind of the key event relationships, if you could call it, that would initiate certain measurable endpoints, which are the definition of the key events. And especially with this case study, it's mostly reliant on current understanding, general understanding from scientific literature on the processes, and this is for neurodevelopmental field is quite an issue, quite a challenge. Within the VHP4Safety project, we're now working on scaling it up, improving the methods. So also the generation of these models to make them more consistent and more reliable, also easier to share. So working on a couple of services with user interfaces as we do in VHP. Also exploring in TXG map project different ways of mapping genes to key events, not using just the molecular pathways from Wikipathways, but also other pathway databases and ontologies. And also generating a more automatic pipeline to analyze the omics data, starting from the log2 fold changes, because we have already services available for the processing and quality control, but then working more towards the interpretation of the data. And that finalizes my presentation. I would be happy to answer some questions.
Thank you, Marvin. A nice, your last point is actually a nice segue to the next webinar that we will have, which will be about the IT platform. So it's nice to have a little bit of this teaser in there. Are there any questions for Marvin? You can put it in the chat. You can raise your hand. You can whatever you want. Okay, I don't see any right now. Do we have any from the audience, the other panelists?
And it was expected, yeah, quite clear then, I guess. And let's save some time for Annika, that we don't take 90% of the 1.5 hours.
Yeah, maybe what we will do then in light of time is if you have a question for Marvin, put it in the Q&A or in the chat, and we can address it after Annika's presentation. If that works for you, Marvin?
Sure, yeah.
Yep. All right, then Annika, the floor is yours.
Here, this one.
I'll just
you can
sorry,
see.
I'll just take a quick one. You can leave your presentation up, Annika, but there was a question that came right now. So
Okay.
from Chris, a lot of work was done to annotate inhibition and activation in MIM and SBGN. Is that not usable?
Yeah, on the genes and pathway processes, yes, and that's also part of the molecular pathways themselves. It's just that on the level of the key event, so if we want to say a key event is active or is not active, we would also want to know how the gene expression actually relates to that. So whether certain genes are expected to be up or down regulated. And that's something that I'm not sure is solvable at this moment, but at least we're trying to include like key regulatory genes and understand how they fit into the biology and certain process activation. So that is a way to go, but we're still exploring how to do this most effectively for the whole pathways that we are interested or that are linked to those key events. It's a fair question. Yeah, I don't have the very clear answer. Sorry.
Thank you. So if there are more questions, please feel free to type them in the Q&A. And Marvin, you can also type an answer to them if you would like to. And now Annika, for real.
Okay, great. Yes. Yeah, then I will start. So yeah, I'm Annika Drees. I'm a PhD student at the VOB, and I work in ONTOX on the investigation of AOP-anchored events of chemical-induced cholestasis. So yes, a quick overview of my talk. I'll first introduce you to chemical-induced cholestasis and tell you about the optimization of the AOP network that has been done, then introduce you to the human-based cell culture model that we're using, and then show you in the ONTOX liver cholestasis case study some methods and results that we've achieved, and at the end some conclusion and future perspectives. So chemical-induced cholestasis is an interesting area to look into due to two main issues. So for one, we have a high incidence. So if you just look at pharmaceuticals, it's a common and potentially very severe adverse effect, with numbers stating around 40% of all cases of drug-induced liver injury being of cholestatic nature, and up to 8% of those being fatal. And it doesn't just stop at the pharmaceutical compounds. There are also known food additives, cosmetic ingredients, and industrial chemicals that cause cholestasis. And the second part of the problem is the identification, as we currently have very poor predictability of chemical-induced cholestasis with the methods that are being used during drug development and risk assessment. Then, as we know, animal studies like the human relevance and simplified in vitro models, they focus on maybe one or two aspects of cholestasis, but then fail to express the full mechanism and therefore miss a lot of inducers. So the solution that we are looking at is the use of an AOP-based in vitro test battery, which means you would first need to unravel the mechanism behind chemical-induced cholestasis, select an appropriate human-based in vitro model to have this human relevance, and then target multiple points in the mechanism to hopefully include the whole mechanism of cholestasis. So for the optimization of the AOP network, this was done by my predecessor. So the initial AOP network for chemical-induced cholestasis was set up in 2013. But in over a decade, there was a lot of new information generated, and this should be integrated into the existing AOP network as well. So in 2023, they decided to optimize it. The workflow is here shown on the right, and taken from Jonas van Erdvelde's publication. I will not go into all the details, but this was an AI-assisted optimization. So what they did, they collected a lot of articles from PubMed that fit certain literature search. They screened these abstracts using Sysref as well, with 10% of the abstract being screened manually and 90% of them with AI. This was done again, and the data was extracted and analyzed from those articles that fit the criteria. In the end, it resulted in 135 unique key event relationships, or KERs, and 38 unique key events. And they didn't only want to include the type of key event relationship, they also wanted to add a weight to them. So what they did for the key events, they looked into the incidence, so how often a key event is mentioned inside an AOP, mentioned inside the database. And for the key event relationships, they made use of the tailored Bradford-Hill criteria, which was set up by the OECD. So you looked into the empirical evidence, so dose and time concordance, the essentiality, and the biological plausibility, so maybe uniformization of dose, so how uniform the data is, which resulted in a total key event relationship score. And both the incidence and the score were visualized in the AOP network via the node size and the edge size, but I will show you in more detail here. So this is the simplified AOP network that resulted from it. Simplified meaning, it is summarized, not all 38 key events and 135 key event relationships are shown here. What we see is that we have the molecular initiating events, which are here in blue, and are the transporter changes, biochemical changes, and hepatocellular changes. Then the key events, which are in this yellow-orange, which are split into the adverse response and the adaptive response, and they all in the end lead to cholestasis. And as I mentioned, the larger the square, the more often this key event was present in the data set, and the thicker the arrow, the more confident we are about this key event relationship. So then we had to select a human-based cell culture model that we could use to test the AOP network, the AOP anchored events. So we decided to go with 2D cultures of Hepar-G cells. These are human origin cells, and they are very well established and characterized. I think they're seen as one of the best models following primary human hepatocytes, since they express both phase 1 and phase 2 enzymes. They also functionally express transporter proteins, even some of them at this correct location. And they also form these bile pockets, since this is a combination of hepatocytes and cholangiocytes. And on the image on the right, you can see these bright white spots. Those are these bile pockets that are being formed by the cells. And then for the ONTOX liver cholestasis case study, we had to focus on some our targets. We couldn't test all of them, of course. So we decided for the MIEs to go with the cholestasis-specific MIEs of transporter changes and biochemical changes. And for the key events, we're looking into adverse response key events of endoplasmic reticulum stress, oxidative stress, and mitochondrial impairment. So mitochondrial impairment is not part of the simplified AOP network. It's still part of the AOP network of cholestasis. And it's also for the other liver case study that we have of steatosis is an important key event. So it was still interesting to include here to have this comparison. So I will now go into a bit of the testing of it. So if we look into the transporter changes, the transporters relevant for cholestasis can be split into 3 groups. We have the ones that are responsible for the basolateral uptake, so from the blood into the hepatocytes, then basolateral efflux from the hepatocytes into the blood, and then canonical efflux from the hepatocytes into the bile caniculi. And we test those for one transcriptionally, so we investigate the changes of the gene expressions of genes that encode the transporter proteins after exposure to our compounds using RT-qPCR. But then we also test functionally, where we use fluorescent substrates of the different transporters, and then just can measure the change in those fluorescent signals to see if there's any inhibition. So basolateral uptake, we have a probe that targets OATP1B1 and OATP1B3. Basolateral efflux, we target MRP3, and canonical efflux, we target MRP2. For the first two, it's very simply measured in the wells via a plate reader, and we just see if there's a change in the fluorescence intensity. Canonical efflux is a bit more complicated, but also a bit nicer to look at. So what we do is we expose ourselves to the compounds and to a compound called CDFDA, which is then passively uptaken into the cells, metabolized, and then actively excreted into the biocaniculi as the fluorescent CDF. On the left side, you see our vehicle control, such as DMSO, and you can clearly see these like bright green bile pockets that have been stained with CDF because they were efflux via MRP2. But then on the right, we're using entacapone, which is our positive control for this assay, and it inhibits the transport. And instead of these brightly colored biocaniculi, you have the signal inside of the cell because the efflux is inhibited. And what we do is we take these images after exposure and then use semi-automated image analysis to count the number of stained biocaniculi and compare the control to our exposure conditions. For the biocanicular changes, we test those functionally and we look into the biocanicular dynamics. So these biocaniculi, they can, the normal condition, they are pulsating, so dilatating and constricting constantly. But if you add a cholestatic compound, what can happen is that you lock them in one of those states constantly. So constantly dilatating or constantly constricting to the point where they could even disappear. So what we do is we use a 6-hour phase contrast microscopy. So we add our compounds to the cells, and every hour we take an image. And at the end, we use again the semi-automated image analysis to quantify the area and the number of biocaniculi that are still left in the wells, and compare again to the control condition to see if there was a change. And here on the right, you see a little video of the time lapse. So every hour an image, and here we use MK571, which is our positive control for this assay. So then we look into our adverse response key events. These are also tested functionally. And what we do is we target specific adverse response related endpoints from each key event just using fluorescent probes. So for endoplasmic reticulum stress, we have a probe that targets the protein aggregates. For oxidative stress, we target reactive oxygen species. And for mitochondrial impairment, we look into the mitochondrial membrane potential. This is also very simply analyzed. So we use the fluorescent probes and measure inside of the well and just see if there, via a plate reader, to see if there's a change in fluorescent intensity compared to the control. So this is not, I don't have any nice images, but I can show you an example of the type of results that we achieve. So here, this is the mitochondrial impairment. We use the probe rhodamine-123. This probe is taken into the mitochondria relative to its potential. So if you have mitochondrial impairment and therefore a loss of mitochondrial membrane potential, you will have a decrease in signal. And here on the left, you see the compounds that we are testing. So we use 5 non-causatic compounds with ANIT, PFOS, cyclosporin A, ticlopidine, and chlorpromazine, and 2 non-causatic compounds with acetaminophen and propanolo, testing in 4 different concentrations. As you can see here, we have a positive signal with the positive control of FCCP, ANIT, ticlopidine, and also propanolo, which is supposed to be non-cholestatic, but since these adverse response key events are not cholestasis specific, we do expect some sort of more or less false positive results. So now at the end is the question, how do we analyze everything? Because the idea is that we derive the cholestatic hazard from the entirety of the test battery outcome, not from individual assays. So what we want to do is to use a weight of evidence approach, which means we assign a weight of to each different measurement. Again, similar to what we did for the AOP using the Bedford-Hill criteria and the assay performance. On the right, it's like a lot of information, but this is the basis for the weight of evidence approach that has been recently published by my great colleagues Amy Merton. This they tested the molecular initiating event of cholestasis, as you can see in the top row. So the different transporters functionally and transcriptionally, as well as biomolecular dynamics as well. And each of these measurements was assigned a weight based on the Bedford-Hill criteria, which is in the row below. And as you can see that the biomolecular dynamics was given the highest weight. And here, how do you use this table? So every time a compound was statistically positive in an assay, it is given a 1. If it's negative, a 0. You multiply this number by your weight of evidence score for this specific measurement. At the end, you sum up all the numbers, and then you have here on the right side your sum of all of them together. And the idea is that you can define a cutoff point in which you can differentiate between cholestatic and non-cholestatic compounds. And you can see here it's already very promising results, as the bottom 4 are negative controls, and they generally have more or less lower numbers than the positive controls. So just for the conclusions and future perspectives, so we were able in our case study to successfully target the MIEs and key events in the AOP network of chemical-induced cholestasis with our assays. We have also almost completed the full testing of the pilot compounds in the in vitro test battery. I'm not able to show you all the results. We would have been here a while. But generally, we have promising results in the performance of the test battery and the detection of cholestatic compounds. We have already also a basis for the weight of evidence approach available, which we do need to expand upon, as we're now also including the adverse response key events, as well as multiple concentrations per compound. Also, as you might know, ONTOX isn't only in vitro work. We also have some great in silico groups working with us. So we also want to include in silico approaches as well, more specifically QSAR modeling and docking models. So at the end goal, we would have an integrated in vitro and in silico test battery for the improved prediction of cholestatic hazard of compounds compared to what we have now. And with this, I want to thank you for your attention, and I want to give a special thanks to my promoters, Matthew Winken and Julian Sanserano, Amy Merton, the other great colleague who works on cholestasis, and Anouk Verhoeven, who works on a steatosis case study in cholestasis, in our team, sorry. And also the rest of the HepX team, of course, and ONTOX. So thank you very much.
Thank you, Annika. There are already questions in the Q&A. First one from Chris. How do you count semi-automatically? I'm not sure what this refers to, but
yeah, this is we use the software ImageJ, and it's semi-automatically because we first have to set up a macro that runs to count the cells. But after we've done it for like one image, we can then run them through all of our other images as well. So it's not fully automatic, but still has a lot of manual input as well.
All right. And then we have from Susanna, PQ, which I assume is paraquat, shows no effect on the tested transporters. Is it known if the mode of action for cholestasis is very different?
For paraquat, there's the interesting example. We don't have much data that it's not in humans. So paraquat, there's a lot of case studies available, but there's not much done in like in vivo and in vitro studies. So there isn't much known about the mechanism at all.
Okay, so
yeah,
it's
so it's a mystery, I guess.
Okay, all right, thanks. Are there more questions from the audience in the chat, in the Q&A? Raise your hand. Give it a minute. In the meantime, did we have all the questions answered from the previous talks from David and Marvin? I think so, yeah. All right, well, if there are no burning questions, then we're actually, oh, wait, one. From Susanna again, would the measurements of the bile acids accumulation help to understand or even confirm the cholestatic potential?
Yes, there is the idea to, yeah, bile acid accumulation is like a very important key event, but we found out it's not that easy to assess. So we were a bit split between like the high throughput compatibility and like the performance. So we have decided to now exclude it. But there are other people in the team who work also on working on this bile acid accumulation as well. So.
All right. Thank you. If there is nothing else, then I will thank everyone for joining on a Friday afternoon. I hope to see you all at the next webinar, which will be about the IT platform and will most likely be in September after World Congress, Eurotalks, all of this. So keep tuned. We'll promote it on LinkedIn again and within the projects. Thank you to all the presenters for making time for this. Well, thank you to ONTOX also for the support in organizing. They're the silent partners here. And yes, you have a nice weekend. See you all in September. Cheers.
Thank you.
Thank you. Bye.
Bye.